About plasma membrane. Every cell contains a membrane around itself that acts as a protective barrier, and it lets outside stay outside and inside of the cell stays in, the content stays inside unless transport happens across the membrane. The membrane contains phospholipids, and because they are in two layers, we call it phospholipid bilayer. The green heads, the blobs, are made up of a phosphate group and glycerol, and they are hydrophilic, so they like the water. On the other hand, the tails of the phospholipids contain two fatty acid tails, and they are hydrophobic, that they don't like water. The big blob over there is a protein molecule, a channel molecule that let different materials go in and outside from the cell. What I'm drawing is the cholesterol molecules or in the cell membrane that they act as stabilizers. On the surface, they have some sugar chains that act, or carbohydrate chains, that they act as receptors or the communication tool between cells. The arrows indicate that these proteins don't stay stable inside of the plasma membrane. It's kind of a fluid mosaic model. They can move left and right. So the function is the plasma membrane is a barrier, and it's selectively permeable. What does it mean, selectively permeable? It means that it lets some materials go through and some materials not. Like water solubles cannot pass through because of the hydrophobic layer. On the other hand, fat solubles can go through. And large molecules, not really, unless they have a channel, like the protein channel, that let them go through. Depending on which direction they go, we distinguish two types of transport for the molecules. It can be passive transport or active transport of the molecules across the phospholipid bilayer. The passive transport is driven by concentration gradient along the membrane, and it goes from high concentration to low concentration. On the other hand, the active transport is driven by energy, ATP, and that goes from low concentration to high concentration. The other important topic what we have to cover with the cell membrane is the tonicity. Let's assume we have a glass of water. Inside of this cup is water. That to the water we add 1 tablespoon of sugar or salt, something really that would dissolve in the water. In this case, the water is the solvent, and the sugar is going to be the solute that gets into the water, into the solvent. Together, the solute and the solvent makes up the solution, that a homogeneous mixture. In scenario 1, we will add couple of extra teaspoons of solute, sugar or salt, whatever you think about, to the water, to the solvent, to make a thicker solution, because the solute is more now in the solvent. It's a hypertonic solution. Scenario 2, let's dilute that thick liquid, sugary liquid, with water, a gallon water or 2, so it will be very less sugary. The concentration of the solute would be less, way less than it was before. We call it hypotonic solution in this case. And we will use that later, this knowledge.